Medicinal activities of curcumin, from ancient remedy to modern drug discovery. Curcumin, the principal bioactive polyphenol isolated from the rhizomes of turmeric, Curcuma longa, is one of the most extensively investigated natural products in modern medicinal chemistry. For more than 5,000 years, turmeric has held a central place in traditional Indian Ayurvedic medicine, where it has been used for wound healing, gastrointestinal disorders, inflammation, skin diseases, and blood purification. Similar medicinal applications were documented in traditional Chinese medicine, making turmeric one of humanity's oldest therapeutic agents. The bright yellow spice eventually reached Europe via ancient trade routes and later attracted scientific attention for its remarkable biological activities. Discovery and chemical structure of curcumin. Modern research on curcumin began in earnest during the 1970s, when investigators reported its cholesterol-lowering effects in experimental animals. Interest expanded dramatically in the 1990s with growing recognition that chronic inflammation plays a crucial role in cancer, cardiovascular disease, neurodegeneration, and autoimmune disorders. Curcumin emerged as a promising anti-inflammatory molecule capable of modulating numerous cellular signaling pathways involved in disease progression. Chemically, curcumin is a lipophilic diaryl heptanoid consisting of two methoxyphenol rings connected through a conjugated beta-diketone linker. This unique structure enables antioxidant activity through hydrogen atom donation and radical scavenging, while also allowing interactions with multiple molecular targets. Unlike conventional drugs designed to bind a single receptor, curcumin exhibits pleiotropic activity, influencing transcription factors, cytokines, growth factors, kinases, and inflammatory mediators simultaneously. Curcumin as an anti-cancer agent. One of the major milestones in curcumin research came from studies linking inflammation to cancer development. Curcumin was shown to suppress activation of nuclear factor kappa B, NF-kappa B, a transcription factor that regulates genes involved in inflammation, cell proliferation, angiogenesis, and metastasis. In addition to NF-kappa B, curcumin modulates several cancer-related pathways, including STAT3, EGFR, TNF-alpha, and P53 signaling. Laboratory studies demonstrated that curcumin could inhibit tumor growth, induce apoptosis, suppress angiogenesis, and reduce metastatic potential in numerous cancer cell lines. Therapeutic applications beyond cancer. Curcumin has also shown therapeutic promise beyond oncology. Experimental and clinical studies have explored its potential in arthritis, inflammatory bowel disease, diabetes, cardiovascular disease, metabolic syndrome, Alzheimer's disease, and neurodegenerative disorders. Its potent antioxidant and anti-inflammatory effects are believed to underlie many of these benefits. Clinical trials have reported reductions in inflammatory biomarkers such as interleukin-6, IL-6, interleukin-8, IL-8, and tumor necrosis factor alpha, TNF-alpha, supporting its role as a disease-modifying agent. The Bharat Aggarwal controversy and its impact on curcumin research. Despite these encouraging findings, curcumin research has experienced significant controversy. One of the most prominent early advocates of curcumin as an anti-cancer agent was Bharat Aggarwal of the University of Texas, MD Anderson Cancer Center. Aggarwal published hundreds of papers promoting curcumin's therapeutic potential and helped popularize the concept of targeting inflammation to treat cancer. However, subsequent investigations uncovered extensive image manipulation and scientific misconduct in many of his publications. As of 2021, at least 29 of his papers had been retracted. These revelations raised concerns regarding the reliability of some early curcumin studies and fueled skepticism within the biomedical community. Importantly, the misconduct associated with individual researchers did not invalidate the broader body of curcumin research. Independent laboratories worldwide have continued to investigate curcumin using rigorous experimental methods. The scientific consensus today is more nuanced. Curcumin undoubtedly exhibits diverse biological activities in vitro and in animal models, but translating these effects into clinically effective therapies remains challenging. Curcumin analogues, synthesis and enhanced biological activity. The greatest obstacle to clinical application is curcumin's poor bioavailability. Oral curcumin suffers from low aqueous solubility, poor intestinal absorption, rapid metabolism, and rapid systemic elimination. Even gram-level doses often produce only nanomolar concentrations in human plasma. Furthermore, curcumin undergoes rapid degradation under physiological conditions, generating metabolites such as ferulic acid, ferulic aldehyde, and vanillin. To overcome these limitations, medicinal chemists have devoted considerable effort to developing curcumin analogues with improved potency and pharmacokinetic properties. One common strategy involves modifying the aromatic rings or central diketone linker to increase chemical stability and target affinity. Monocarbonylic curcumin analogues, in which the unstable beta-diketone moiety is replaced with more stable linkers, have demonstrated significantly enhanced anticancer activity. Some synthetic derivatives exhibit tumor growth inhibition up to 30 times greater than native curcumin. Another successful approach involves replacing the guaiacol rings of curcumin with heterocyclic or coumarin-based structures. Fluorinated coumarin curcumin analogues have shown superior antioxidant and anti-tumor activity, while chlorinated analogues display enhanced antibacterial effects. Structural modifications that increase hydrophobicity or introduce electron-withdrawing substituents often improve cellular uptake and biological activity. Nanotechnology and advanced drug delivery systems. Metal-complexed curcumin derivatives have also attracted attention. Coordination of curcumin with copper, ruthenium, zinc, or platinum ions can improve stability, cellular uptake, and anticancer efficacy. In parallel, nanotechnology-based formulations, including liposomes, micelles, dendrimers, hydrogels, and polymeric nanoparticles, have dramatically improved curcumin delivery. Commercial formulations such as phytosomal curcumin and liposomal curcumin produce significantly higher plasma concentrations than conventional extracts. Co-administration with piperine, the active component of black pepper, can increase curcumin bioavailability by as much as 2,000%. Future perspectives and conclusions. Today, curcumin occupies a unique position in drug discovery. It is neither the miracle cure once claimed by some proponents, nor the ineffective compound suggested by critics. Instead, it represents a valuable molecular scaffold that continues to inspire medicinal chemists. Advances in synthetic analogues, metal complexes, and nanomedicine are transforming curcumin from a traditional herbal remedy into a platform for developing next-generation therapeutics. Although significant challenges remain, the enduring scientific interest in curcumin reflects its remarkable chemical versatility and its potential to contribute to future treatments for cancer, inflammatory diseases, and other chronic disorders.